Unveiling the Golgi Apparatus. Journey into the heart of the cell's intricate machinery. The Golgi apparatus, a vital organelle in eukaryotic cells, plays a crucial role in processing and packaging proteins and lipids. Structure. The Golgi apparatus is a membrane-bound organelle found in eukaryotic cells. It was discovered by Camillo Golgi in 1898. Structurally, it consists of flattened sacs called cisternae, arranged in stacks. The cis-face receives materials from the endoplasmic reticulum, ER. The transface modifies, sorts, and exports materials to different destinations. It modifies proteins and lipids by processes like glycosylation and phosphorylation. It packages molecules into vesicles for secretion or use within the cell. It plays a key role in the formation of lysosomes. In plant cells, it helps synthesize cell wall materials like pectin and hemicellulose. It functions like the cell's post office, processing and dispatching biomolecules. The cell's post office, an introduction. Also known as the Golgi complex or Golgi body, this sophisticated organelle is an indispensable component of the cell's endomembrane system. It acts as the central hub for modifying, sorting, and packaging macromolecules. Imagine it as the cell's highly organized post office, ensuring that every protein and lipid reaches its correct destination, whether within the cell or for secretion outside. Architectural precision, the Golgi structure. Flattened sacs. The primary structural units are cisternae, flattened, membrane-bound sacs. These are stacked together, forming distinct functional compartments. Connecting tubules. A network of delicate tubular structures interconnects the cisternae, facilitating the passage of molecules between different compartments. Transport vesicles. Small, spherical, membrane-bound vesicles constantly bud off from and fuse with the Golgi, acting as shuttles for molecular transport. Beyond packaging, the Golgi's diverse functions. Protein modification. The Golgi actively refines proteins synthesized in the endoplasmic reticulum, adding intricate carbohydrate chains, glycosylation, and lipids, which are crucial for protein function and targeting. Molecular sorting. It meticulously sorts and packages proteins and lipids into vesicles, directing them to their precise intracellular destinations or preparing them for secretion from the cell. Lipid synthesis. Beyond processing, the Golgi also synthesizes specific lipids, such as glycolipids and sphingomyelin, contributing to cell membrane integrity and signaling. Golgi diversity, adapting to cell needs. The Golgi apparatus exhibits structural and functional variations depending on the cell type and its specific requirements. Supranuclear Golgi. Found in polarized cells like epithelial cells. Positioned above the nucleus, indicative of directional secretion. Optimized for vectorial transport of proteins and lipids to specific cell surfaces. Randomly distributed Golgi. Common in non-polarized cells such as fibroblasts. Scattered throughout the cytoplasm, reflecting less directional secretion. Supports generalized trafficking and processing functions without a dominant secretory axis. When the Golgi fails, diseases and disorders. Dysfunctional Golgi apparatus can have profound consequences, leading to a spectrum of debilitating diseases. The intricate processes of the Golgi are susceptible to disruption, and when they go awry, the impact on cellular health can be severe, contributing to a range of pathologies. The pathological impact. Cancer. Aberrant glycosylation patterns, often originating in the Golgi, are hallmarks of cancer cells. These altered sugar structures can influence cell growth, metastasis, and immune evasion. Neurodegenerative diseases. Emerging research points to Golgi dysfunction as a contributing factor in conditions like Alzheimer's and Parkinson's. Impaired protein trafficking and accumulation of misfolded proteins can lead to neuronal damage. Genetic disorders. Mutations in genes crucial for Golgi structure or function can result in congenital disorders of glycosylation, CDGs, a diverse group of metabolic diseases affecting multiple organ systems. Innovating solutions, restoring Golgi function. Researchers are actively pursuing novel strategies to combat Golgi-related disorders, leveraging advancements in molecular biology and therapeutic development. Therapeutic targeting. Developing precise drugs that modulate specific Golgi-related pathways or enzyme activities to correct molecular imbalances. Gene therapy. Utilizing gene editing techniques to correct underlying genetic mutations that compromise Golgi integrity or function, offering potential cures for inherited disorders. Cellular reprogramming. Exploring methods to reprogram diseased cells to restore normal Golgi architecture and functional capacity, potentially reversing cellular pathology. Key takeaways: the Golgi's enduring importance. Central hub: the Golgi apparatus is a pivotal organelle for protein and lipid modification, sorting, and packaging in eukaryotic cells. Structured for function, its unique structure of cisternae, tubules, and vesicles facilitates complex biochemical processes. Cellular health guardian: proper Golgi function is essential for maintaining overall cellular homeostasis and preventing disease. Therapeutic frontier: understanding Golgi dynamics offers promising avenues for developing treatments for a range of human diseases. Future directions: advancing Golgi research. The continuous exploration of the Golgi apparatus promises deeper insights into fundamental cell biology and new opportunities for medical intervention. Next steps for research: high-resolution imaging of Golgi dynamics in live cells. Identifying novel Golgi-associated proteins and their roles. Developing personalized therapies based on individual Golgi dysfunction profiles. Your role in discovery. As aspiring biologists and researchers, your curiosity and dedication will drive the next wave of discoveries, unraveling the remaining mysteries of this fascinating organelle. What questions does the Golgi spark in you?